So data preprocessing starts here with the count table, and you have something called the metadata table. So the metadata table is information that relates to your experiment, to your experimental setup, variables that you might be interested in. It's generally a good idea to include all the variables that you might know about that are related to the experiment, because they're not just useful for the analysis, but they're also useful for like troubleshooting or for QC steps, or yeah, even in situations where you might think there might be something wrong. It's good to have all your metadata organized into a table, something like this. So you have your genes here, and you have your samples, and in your metadata table you have your samples here along the rows. So the samples here must match your samples here in the count table. It should be identical and in the same order. So once you have this, then you are ready to go, ready to actually start with the pre-processing step. So,
sorry, we have a question from Jacob already.
Yes.
For the previous lectures or presentations, we could download the file as a PDF, but this file is a HTML. Will it be converted into a PDF, or should we take care of it ourselves?
So I think if you go to this link and the last bit here, if you change the .html to .pdf, I think it should work.
Okay, cool.
Yeah, now you have a PDF. Okay, so we have the count table and the metadata. And the first step is filtering. So we want to filter out genes that are not useful, or genes that are bad, or genes that are not like they don't really add value to downstream steps. And the most obvious case is a situation where you have genes which are just completely zero across all your samples. So this is like, yeah, it's totally pointless. So you might as well just remove them. If you're using a package, then this is actually performed within the package. So it's not like super important that you do it, but I generally do it. And it is, I think, good practice to remove bad genes right at the beginning. And there are many ways to do this and many different strategies to go about doing this. So here are 3 different ways to do this. Here, these figures are actually just showing like total count distribution across all your samples and across all your genes. So this is how your raw data looks like. Here, the very first figure. And then here you have method 1, which corresponds to the very first line here. And then you have method 2 and 3. So these are like 3 different ways, or they're not really, all of them are not different, but yeah, 3 ways in which you can perform this filtering step. So if we kind of like, this might look a little bit complicated if you're not like super familiar with R, but if you try to kind of break down this, so the first step here is you have CR is just a variable for row count, or it actually looks like this. So this is your row count. And when you do row counts greater than 0, then every position here, if it's greater than 0, becomes true, and otherwise it is a false. So here, the first row would be, for example, false, 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 false. This row would be false, true, true, and so on. And then if we go to the next step, it's row sum. So all the trues and false, you sum them up row-wise. So in R, true, if you sum up true and false, true is considered as 1 and false is considered as 0. So if you sum up all of this, that would be, yeah, it would be 1, 2, 3, 4, 5. Yeah, that would be 0 basically. And here you would have one sample which is above 0, second one, 3, and 4. So this row would sum up to 4, and so on. So that is what this step does. And then the next step is basically this whole thing. And that is, again, a conditional statement where we are asking, so the total sum, is it greater than or equal to 3? And why 3 is just because we have 6 samples, and we know that 3 of them belong to group A and 3 of them belong to group 2, or group B. But that depends on your experimental setup and your sample and so on. Whether this should be 0 or whether this should be 3 or some different value, again, depends on your particular data set. So this is kind of subjective to your particular sample and your particular setup. The third option here is a bit less subjective, but it's still kind of, yeah, depends on the data set. In this case, we are first converting the row counts to counts per million. So yeah, we're controlling for like the sequencing depth difference in the total number of sequence reads per sample. And then we are saying, okay, if the count per million is greater than some number, then it's a true. And then the rest is the same. So the way to go about doing this is to like try this out. It's a bit of an iterative process. You try it out, look at these distributions and see that they are looking, yeah, they're not looking too weird. Like if you see like a huge peak here, that means there's a lot of low counts. And the first figure here, there's this massive spike here on the left because of all zeros. So there's a lot of zeros in the data. And then we are removing a lot of genes that are low counts and zeros. And then we eventually end up with a distribution that looks something like this. So this is the whole point here. And as you do this, you want to keep an eye on the total number of genes that are remaining in your data set after filtering. Because this is, it's a sort of balance between getting rid of, yeah, poor quality genes. At the same time, you don't want to get rid of too many of them. Because if you set up a filter where you just remove, I don't know, 90% of the genes, then you're losing a lot of information. Again, depends on the data set. I mean, if 90% is really bad, then yeah, it's probably better to keep the better ones. So it depends. So in this case, yeah, we start with 55,000 genes, and then we kind of, yeah, decide to go with one of these methods, and this is how different they are after filtering. Yeah, many questions?
I had one question, and then the 3 is related to your biological replication, because in this study was 3, you used 3, or is some other reasons for the number 3 in your comments?
The 3 is, yeah, like okay, so if you said, okay, greater than equal to all your samples, then the problem is that if some genes are like, you know, completely turned off in one experimental conditions, then you lose those samples or those genes. If you don't set, because if you set it to like all the total number of samples, like if there are 6 samples here, so if you set this to 6, you're being too stringent. So 3 is just to relax the stringency a bit, so we don't lose too many genes. It could go down a bit more, maybe 2, if you want to be a little bit more relaxed. But 3 seems to be a reasonable number because it's there are 2 groups and each group has 3 samples. So we are saying that these genes should be expressed in at least one group. That's what this means.
Another question from Matus.
I have a similar question. Is the number 5 for CPM generally accepted in the community, or it's how to take home message from these?
So normally you want to have at least one count per million. That is the bare minimum, at least one count per million reads. Is that how it is, Paulo, or should it be a bit more than that?
No, that will be enough, yes. But here in this example, the thing is that these samples were already downsampled for 1 million reads. So if you put CPM above 1, that will not filter anything because we have less than 1 million reads. But in general, yes, above at least 1 CPM.
Mm-hmm. And if you read through some of the documentation from, like, for example, DSIC2 or one of these vignettes, this is the sort of approach that they use because it's a bit less subjective. But this 5 depends on your data set. Usually 1 works. Mm-hmm. But you, plot out your counts and then see how they look like, and then you can decide if you want to adjust that a bit. Yeah.
Yeah, thank you.
Okay, so once you have your filtered counts, then we can go to the next step. Well, the next step, yeah, I will talk a bit about normalization and the different kinds of normalization and what are they for and what they do. And then we will talk about the practical aspects of normalization. So this is just a bit of a theory here. So why do we do normalization? So the very, very basic thing you need to do is controlling for sequencing depth. So this is, let's say, a situation where you have pooled multiple samples together. And of course, at the library prep stage, you try to maintain equal quantities of library within the pool, but it never comes out perfectly equal. You always have a few samples that are a bit more or a few samples that are a bit less. So you never get identical number of reads across samples. And therefore, the row counts, you cannot directly compare the row counts because the total number of reads that you sequence are different. And you need to control for this. This is called total count normalization. If you just control for the sequencing depth, and this is super easy because so you have in this example here you have 3 genes X, Y, and Z, and you have 2 samples A and B, and basically you just divide each of this by the total number of reads for that sample, and then you get total count normalized reads. And then this should be comparable. That's the idea here. So total count normalization is, well, it's just important that you know it, but in practice we never just do total count normalization and use those rates because you need to do a bit more. So the next sort of normalization is controlling for compositional bias. So the idea behind compositional bias is that if you have a few genes which are super highly expressed in one condition, so in this case you have a gene here which is extremely highly expressed in one condition, or let's say one sample, and in the other one it's extremely low. In this case, most of the reads are going to be skewed by this super high expression here. Or the other way you can think about this is, if you think of this as a pie, a very large proportion of the pie is made up of these reads from this particular gene because this is highly overexpressed. And then the proportion of the other genes will be much smaller compared to this one. So this is what is called a compositional bias. It's a bias due to overexpression of one or few genes. And this makes it harder to compare one gene to another gene or between samples. And total count normalization does not account for this bias. So total count normalization wouldn't work for this sort of a scenario. So this is just to show you what this is. But we will talk about methods for correcting compositional bias in the next few slides. So in this presentation, if you press on P as in Papa, I have added some text which may or may not be helpful, but you're welcome to read through them in your own time. Yeah, some explanation for what you see here on the plot, on the slides. Okay, so that is sequencing depth and compositional bias, and the next thing is actually controlling for gene length. So usually you don't need to do this because you're comparing one gene in one sample to the same gene in the other sample. In that case, it doesn't really matter whether you control for the length of the gene, because you're never comparing one gene to another gene. If you do need to compare one gene to another gene, then you need to control for the length of the gene, because longer genes, they tend to produce more reads than a shorter gene. So controlling for gene length is basically just taking your reads and dividing it by the length of the gene. That's it. So this is again in theory, but in practice, it's there's a bit more to it, and we will look at some examples later. But this is the idea about controlling for gene length. When you use one of these packages, D62, Edge, or Lemma, whatever, they don't control for gene length because it's not necessary for what they are doing. But if you need to do some analysis of your own where you need to compare gene to gene, then this is something that you need to do yourself. Yeah. And the last point that normalization tries to do is to kind of bring it to a human-friendly scale. This is why we have counts per million. The per million part, or RPKM, the per million part is just makes it easy for us humans to look at these numbers and make some sense of them. Otherwise, they're going to be like 0.000000032 or something, which is a bit difficult. So these are generally the 4 points that we try to control for with normalization. It's not one or the other. It could be controlling for all of these things or some of these things. So these are generally what you want to control for.
We have a question.
Yes.
Okay. But when you say gene length, do you mean the length of the spliced gene transcript or total one?
Yeah.
Yes, it's the processed one, the mRNA length or exonic length.
Yeah, that's a good question. Yeah, by gene length, it's actually transcript length. That is what really matters. So it's all the different exons that a transcript is composed of. So you sum up the length of all the different exons that is part of a transcript, and then you get the total length for the transcript. So one gene can have multiple transcripts, so they could all differ. So this is why, if you read about this, there's a thing called gene or gene length, but there's also effective length. So I think this is what they talk about, effective length, when you look at the papers.
Yeah, thanks.
Mm-hmm. More questions? Okay, otherwise we continue. Yeah, a bit more about normalization and some more details. So in this slide, this slide and the next 2 slides, I'm just kind of grouping the different normalization methods. This is based on this paper, so you're welcome to go and look this up if you feel like. So in this paper, they have tried to kind of group different strategies based on their assumptions. And one of this grouping is to kind of normalization by library size. So here, the primary factor that you're controlling for is the library size. And there are some assumptions when you do this kind of normalization, and this includes roughly, very, very roughly speaking, total count normalization, RPKM, FPKM, and TPM. So total count, as I said, is like, it's the most basic, just controlling for sequencing depth. RPKM, FPKM, and TPM they are related, as in they control for gene length and sequencing depth. Now, you tend to see RPKM and FPKM quite a lot, but they are kind of going out of favor because they are, well, let's look at this table. So we have, again, like three genes, and the last row here is just a sum of the columns on the top. So here we have row counts for A and B, 2 samples, and then we have the length, let's say 20, 2 kb, 4 kb, and so on. And then we have these numbers here. So again, if you press on P, I have written the equations here. If you are kind of interested in going and looking more into this. But these are like the steps if you wanted to do it manually. So you first compute reads per million, and then you divide by the length, and then you get RPKM. And one of the drawbacks of RPKM is that when you sum them up, they don't sum to the same quantity, so they differ. And this means like the proportion of each gene is different, which makes it a little bit hard to kind of compare this 20 to this 11. In contrast, if you look at reads per million or transcripts per million, they sum up to the same total quantity, which makes it more comparable, where the genes are, the normalized counts are more comparable. And this is why TPM is preferred over RPKM or FPKM. RPKM is very similar to FPKM. The only difference is that RPKM is for single end reads and FPKM is for paired end reads, because for paired end you're looking more at the fragment. RPKM, you're just looking at one single end read. The way you calculate RPKM and TPM is very, very similar. It's just there's a very, very small difference in the order in which you do a step. The equation is there, so if that interests you, feel free to go and look it up. But generally speaking, if you are interested in gene length, then preferably use TPM rather than RPKM or FPKM. So in this slide, the only useful normalization here is TPM. You can ignore everything else. Maybe another point is RPM, reads per million. This is the same as counts per million, so CPM. And CPM is usually you log transform as well, so it's log CPM in practice. So the next kind of category of normalization, there are many ways to categorize this, so this is just one way to do it. There are many other aspects that are kind of related, so don't take this grouping too seriously. The next is kind of normalization by distribution. So this is where the correction for compositional bias comes into play. So this is here one of the assumptions is that the technical effects are the same for differentially expressed and non-differentially expressed genes. So if there is some kind of an effect that is not biological, then they should be the same for all the genes. And another assumption is that majority of the genes that you're looking at should be non-differentially expressed. This is an important assumption because many of these methods, all of these methods here, they depend on this fact that vast majority of your genes are not differentially expressed. And in some situations, this assumption might be violated. And one of those situations is if you are comparing, let's say, 2 different tissues, for example. So you have a brain tissue and maybe, I don't know, some other tissue. Expression could be dramatically different between 2 tissues, and a very large number of genes could be dramatically different. And in that case, this assumption might be violated and the results that you get may not be reliable. So that's just something to keep in mind. There are many different methods here with slight variations. So you have Q stands for quantile normalization, you have upper quantile, and you have median. You have many other methods. And two of these methods that I want to kind of mention is TMM and MRN. So MRN is the DSeq2's implementation for compositional bias correction, and TMM is from EDGAR. They are slightly different, but they work in similar ways, as in they control for this shift due to overexpression by a few genes. Because rather than using a total count, or let's say just picking a median, they kind of look at the whole distribution and pick a position that is more optimal for the data that you're actually working with in particular. MRN is, it has a few steps. Again, if you are interested in how MRN works, I have kind of written it down here step by step. And if you want to try it out like in the lab, it's in the differential gene expression lab. So we can look at that maybe tomorrow. So that's a function that runs this, but in practice, you actually don't have to do anything. You just need to know this, because when you run the analysis in any of these packages, it is automatically done for you. So as such, you don't have to do anything. But this is what's happening behind the scenes. And as I mentioned before, this doesn't correct for gene length because it's not useful. Maybe a minor point here is this thing called GETMM, which is gene length corrected TMM. It's not something you see very often, and I haven't really seen it much either. But because if you ask the question like, okay, can we come up with something that is better than, can we have like a normalization that corrects for compositional bias and sequencing depth, but at the same time correct for gene length as well? Then this is that. So it's a TMM normalization which corrects for everything, including gene length. I haven't used it myself, but I don't know. Has any of you guys used it? No?
No.
No. Okay, but it's there. There's a paper, and feel free to go look into it. Questions?
I have one question, if I may ask.
Yes,
so
please do.
you said that FPKM you cannot directly compare if you want to see like, so I cannot claim that gene X is like differently expressed than gene Y. Do I understand it well?
Well, you cannot say anything about differentially expressed.
Yeah.
You can just say, yeah, it's different.
Okay. Thank you.
Okay, there are like a few more categories which they summarize in the paper, but I don't know if it's maybe not like that super important. So there's this normalization by testing. Here, the idea is like, rather than having some sort of an assumption in advance or some sort of a framework in advance, you look at the data and then kind of decide what is the most optimal strategy to go about normalizing the data. And this is what Poisson-seq and this method DGs does. Again, personally, I haven't worked with them, but they are there. And the authors of that paper, they claim that this is a better strategy. Again, it's not widely used, but it does aim to correct some of the drawbacks of the other methods. And in this case, you need to have, it doesn't really work for all kinds of data set. This whole idea came from, I think, one particular experiment where they had a data set which was really hard to work with using the standard methods. And that is how they came up with this alternative strategy. And again, with Poisson-seq, I think the distribution is modeled as Poisson rather than negative binomial. So that is a bit different there as well. Dispersion is not taken into account. So I don't know, may or may not work, depends on the data set, but just so you know, that's there. The next is normalization using controls. So this is a situation where you have some sort of a control gene that you know in advance should be this value, or this is how it should behave across your samples or across the experiment. And this is very similar to how housekeeping genes work in qPCR, for example. So if you know in advance this is how it should be, then you can use that information to say, okay, in this sample, my control is this value, so I expect it to be the same in the other sample, and then you shift everything based on that. And yeah, you can also use spike-ins for this, and you can provide spike-in counts as the normalization, so it plays a part in your normalization factor. So this is another possibility, but then again, it depends on the particular situation in your experiment if you have this. Again, you need to be really sure that your spike-ins are behaving the way you expect them to, because spike-ins are not perfect. It's very hard to measure exact, very precisely equal volumes across all your samples. So there are some practical issues to that as well. Yeah, so that is about normalization. And this last thing here is a bit of a deviation from this normalization methods, but this is something called variance stabilizing transformation, or stabilizing the variance. This is more for exploratory analysis. That's because the other methods, so this is something that's inherent to the data set, as in like the RNA-seq data. It's negative binomially distributed, and you have a situation where there is a mean variance relationship. So variance tend to increase over the mean values. As the mean goes up, the variance goes up much more than what you expect from a normally distributed data. And exploratory methods like PCA has this assumption that the data must be normally distributed and should be homoscedastic, as in the variance should be constant. So if you have heteroscedastic data, then it is violating an assumption. And the idea with variance stabilizing transformation is that you fix this issue so that the data is more suitable for analysis such as PCA or clustering and so on. And DSeq2 has two functions that helps you to create such sort of data, variance stabilized data. And Limma has a function as well, which helps you to do something similar. And yeah, we will look at this during the exploratory analysis. We are going to use these counts there. So that is like a kind of an overview of different normalization methods and yeah, the details of this. The last slide here is basically like the take-home message from all of this, which is when you use a package in R or any package for differential gene expression analysis, the package automatically does all this normalization for you. So what you need to provide is actually the raw counts to the package. So don't do any kind of transformation or yeah, normalization or anything before that in advance. They usually need the raw counts because everything is done internally within the package. And all of these packages, they do a mix of different normalizations. It's not like they pick one or the other. You need all of this it's mixed up in different ways so that it controls for different issues. And then for visualization or exploratory analysis, VSD is preferred. And if you are doing your own analysis where you need to compare gene to gene, then it's important to remember that you should control for gene length. Yeah, a recommendation for that would be TPM, or maybe this new thing called GETMM, probably better because then it's also a control for compositional bias. And there are custom solutions for specific experimental setups where you might be able to use spike-ins or housekeeping genes. Yeah, okay, so here's a link down here, which is kind of a nice overview about overview over these different methods and kind of gives you a lot of details that I have skipped over. So you're welcome to go and look into this to learn more about this. Yeah, so that's really all I want to say with this normalization lecture. Okay, if there are any questions, this is a good time.